Hello, here we are going to review the anatomy of the light microscope. This is a very important tool that we are going to use in our biology lab. Let's go! First, we are going to see the ocular lens or eyepiece lens. It's where the sample is going to be observed. It's the first point of magnification. Usually, it's around 10 times or 15 times of magnification that we are going to observe. Next, we are going to see the body, tube or barrel or head of our microscope. This structure connects the ocular lens to the nose piece. This is a very important piece of our microscope, is where we are going to see the objectives. But we are going to talk about this later. Next, we have the arm. It's called frame as well. Connects the tube with the base of our microscope. It's very important because we are going to use the arm for carrying the microscope to different places. Okay, that's very important. The revolving or nose piece, as I told you before, is where we are going to see the objectives. It's a rotating structure that spin when we want to change in order to get higher magnification. The objectives, very important, is the second point of magnification. Usually we are going to see 4 objectives in our microscopes: 4 times, 10 times, 40 times, and 100 times. In general, there's a color code that help us to identify these objectives. In red, we are going to see the 4 times objective, in yellow 10, in blue 40, and in gray 100. When we want to work with 100 times magnification, we are going to use immersion oil. Next, here we have the stage, is where we are going to put our slide. We are going to observe with the oculars. Our specimen or our sample is here. The slide is attached to the stage by these stage clips that help us to move our sample. It's possible to move the sample in 4 directions using the stage controls or knobs. These 2 structures here are very important because it's the coarse and fine adjustment, or coarse and fine focus, knobs, screw, as you prefer. The screw that is identified with C is the coarse focus that brings us the specimen into a general focus. We are going to use at the beginning with the 4 times magnification in order to see the sample. Then we have this F. This F is the fine focus that we are going to use that when we want to get a very clear image of our sample. Normally we are going to work with this course with the 10, 40 and 100 times magnification. So remember, the coarse adjustment with 4 times magnification and the fine adjustment with the other objectives. Then there's the illuminator or lamp, is the light source of our microscope. It's important because it's a light microscope, so we need light for sure. We can adjust this light using this button here. We can adjust the light using this button here. And there's another option that we are going to see later. Well, under the stage, there's the condenser and diaphragm. Condenser gathers and focuses light from the illuminator onto the specimen beyond view. As you can see, here we have the light, here there's the condenser, and here there's the specimen, the sample. The diaphragm is very important as well because adjusts the amount of light that we are going to get in our sample. We can use the diaphragm, that is more or less here, and this button in order to get less or more light. It is going to depend on the objective that we are using. Okay, for some objectives we need more light, and when we work with another ones we need less light. Finally, here we have the base. The base supports the microscope and is where the illuminator is located. Okay, support all our structure. Well, this is the anatomy, but don't forget to plug in your microscope and turn on the illuminator lamp if you want to work with it. You can see extra information on your notebook in the lab book. You can read pages 9 to 12, okay? That's important. Thank you so much, and I hope this information helps you in the lab. Bye bye.